I'm not going to talk about TrackMate right now. I just want to at least mention this to you because we are now heading into this part of the talk, this entire course thing, which is so we're now heading into the part of the talk which is talking about time-lapse sequences. So how do we track things over time? How do we track movement over time? How do we, I don't know, understand time dynamics in our samples? And now one of the cool tools that Fiji has brought in is called TrackMate. So again, I'm not going to show this to you, but just at least be aware that it exists. Some of the things that it can do is if you've automatically segmented cells, then it can keep track of those cells over time, and then you can create little migration patterns of your cells, for example, that's moving. However you want to do this, I think they even say you can use it with that Stardust thing that I was showing you earlier. It's well integrated into Fiji so that you can, in 3D as well, track movement of different things. Yeah, that's the entire goal why this thing exists, is to track moving objects over time. I just want to see if there are other good examples. So look into that if you want to know more, but we're not covering that specifically. All right, so now we're going to focus on specifically mitochondrial analysis, and then after the lunch break, there will be some cell wound healing type of examples that we will consider. And then Emmanuel he's a guy from Italy, but he lives in Uganda at the moment, so you will be presenting that remotely. Hopefully all of the sound and everything, it works out, and maybe we should just test it just before we start, but yeah, that will be after the lunch break. This is my own joke that I like to present to peers, because I am not a biologist, I'm an engineer. So from an engineer's perspective, in general context, the most that you know about a cell is that, oh, this is what it looks like, because that's what you see in your textbook. And then it's like, okay, great. Mitochondria, where is the mitochondria? There are 4 of them in a cell, okay. They are big blobs that are very easily identifiable. The reality is obviously a lot more complicated than that. They are very highly dynamic, highly networked structures that constantly remodel itself according to the needs of the cell. And what do we have to work with? We only have a matrix of values that is representative of this entire image. And we now want to use this matrix of values to make certain types of conclusions. Now, the one I'm going to talk about a little bit now is where we want to take mitochondrial events. So specifically fission, fusion, and depolarization, just to discuss what this is. In fission and fusion, what I consider fission and fusion is where I've got 2 mitochondrial structures, and maybe let's just define that. A mitochondrial structure is where I've got lots of mitochondria that are already connected together. So they've already formed a network, but they are disconnected from the rest of the mitochondrial network. So then those would be 2 distinct mitochondrial structures. It could also just be a single mitochondrion floating around. That would also be a mitochondrial structure. It could be a little bit of an elongated filament that's floating around. Anything that is separate from anything else, I consider a mitochondrial structure. So I want to know, where do these things, mitochondrial structures in my cell, where do they join? Where do they break apart? Where do they fuse? Where do they undergo fission? And then depolarization is this understanding that before mitochondria basically die, they depolarize. Their membrane potential falls away, and then either the entire cell dies, which is sometimes what happens, or then the specific mitochondria would then be, let's just imagine, engulfed by an phagophore, form an autophagosome, be taken up by a lysosome, eventually be regenerated to components that the cell can use. And I want to know which of my mitochondria, where is this depolarization happening? Is it happening on the periphery? Is it happening in the center? I just want to have a way to automatically detect that. These are the 3 events that I'm trying to get. And my very ambitious desire is, I want to have some algorithm where I can put this single image in, and then I want it to tell me, where is the mitochondria going to fragment, where is it going to fuse, and where will it depolarize? So I want to take this in, get that out as an output image. Okay, now, what is that algorithm that achieves that? Do you have the tools to do that, like what we've discussed now? How would you go about doing this? Going from the one image to another image? To know the location of certain things.
What is the question?
Okay, how do I give my single image into some system, and then out pops the location of where the mitochondria will fuse, where it will fragment, and where it will depolarize. Oh, you can do it with one image. That's actually a little bit of the trick. How do you do it with one image?
Not a time-lapse.
Not a time-lapse. How do you do it with one image? You train a neural network. So you can't do it with one image, like image processing wise, but you can train a neural network to go from one image to another image. How do we train a neural network? What do we need? We need to know, before I get that, okay, so the point is I don't know what the algorithm is, but I just told you it can be a neural network. The big problem here is that we don't have the ground truth. To train a neural network, you need to say, this is the input, this is the output. So I need the input-output pair, and then it can learn how to map from the input to the output. But we don't have this output. This is the thing that we're trying to get. But we can generate it by hand. How do we generate it by hand? Lisa was just giving the hint there. We need a time-lapse sequence. We need a video of our mitochondria, how it's dynamically actually moving over time. And then if we can somehow analyze that, we can generate these images and then ultimately train a neural network on that. So what we did is we captured the time-lapse sequence. I showed you the quick animation at the beginning. We captured lots of them. That was just one. We didn't actually end up using that one, but regardless, it's still in the slides. So we captured this over time. This is in Z, and Z stacks, I mean. And then I can say, this is frame 1, first frame. This is the second frame. And it's since been expanded to even consider more frames, but for now, let's just think of it simplistically. 2 frames, starting point, ending point. What has changed? Structurally, what has changed in my sample? You know, there would obviously be some process flow, but again, how would you do this? How would you even go about solving this? Just think for a moment. Everything that you know, how would I compare 2 frames to then ultimately come to some conclusion? This is where it fused, this is where it fragmented. Say again? No. So this one is not, this one you're not going to train a neural network. This one you actually have to figure out just by manipulating your matrix of values. How would you, given what you now know, how would you define change? Yeah, okay, sure, but tell me the analysis process that you would follow. Like, I would first do this kind of operation, then this operation, and this operation, and then finally I know this is where the values changed. Like, what would that pipeline look like? Okay, so but do I just apply this on the entire image? So where did the events actually happen? There will be some filtering that you would need to do. You would try to get rid of some noise. You would try to get a perfect segmentation. You would try to determine what is foreground, what is background. Once I've got perfect segmentation, foreground, background, I can maybe start to identify where are my individual structures. Once I have the individual structures, the particles, as we've called them before, then I can say, okay, well, maybe how much percentage of this one structure in the one frame is overlapping with these structures in the second frame. So I know spatially where it is. So then I say percentage overlap, for example, between one frame to the other one. And if I've got one structure here that overlaps with 2 structures here, then what probably happened? They fragmented. One structure became 2 structures, so there's fragmentation or fission. If I've got, if I do the inverse, if I say I look from frame 2 to frame 1, I now have 2 structures, and I have one structure in this frame, and then in the previous frame I had 2 structures, so that means that they should have fused to produce that one structure. So I can mostly do this with just looking back and forth between the frames, primarily relying on, let's call it percentage overlap, because there will be some jitter in the mitochondrial network, so you can't just exactly find overlap, but yeah.
In terms of 2 becoming 1,
yes.
how do we know if it's a fusing situation? Would the intensity double or?
No, so usually when fusion happens, there is no real observable change in intensity. It's just that they were 2 separate structures and now they are joined. You'll see some examples just now.
So the size would be bigger?
So let's say I've got 2 filaments, two little worms. Yeah. Then they can come closer and then boom, now they're one worm. That's fusion. Yeah. So the size, you could look at size, that is definitely one component to look at. So if I say in this image, my area for the structure was 100 pixels, and now the area of the structure is 50 pixels, clearly something dramatic happened. This most likely would have been a fission event, but I still don't know where it happened. Like, where exactly did the fission event happen? I don't want to just know that it happened. I want to know where, what pixel is the place where this happened. So there I need to be very more specific. This is the one structure. Now there are 2 structures associated to this one structure. So where between these 2 structures would that fusion point have happened, or fission point have happened? Like you understand, I need two structures to start to consider even where the location is.
Because what if it just depolarized when you lost it? Or
Yeah, if there's something here and then nothing here, then you say, okay, well, in the same spatial area, there was something, now there's nothing, so it must have depolarized.
okay.
So do you do it in 3D?
Yes, you kind of want to do everything in 3D because it's very possible that I've got two mitochondrial structures that's just moving past each other. They never fused, but if I looked at it in 2D, it's going to be, oh, cool, they fused. So, yeah.
Could we apply the localization for that, co-localization for that?
No, not so much. So localization is usually between two different stains, two different structures. So for this, it's just mitochondria. So yeah, not so much for this one. Okay, so let's just, remember pre-processing deconvolution, extremely important because I've got these little structures which are going to come close, and I'll show some problems with this just now. But when they get close, I want to know when they've actually started touching. And if it's a blurry image, it's going to seem like they're touching before they are. Or I wouldn't be even able to distinguish individual mitochondrial structures. It will just be one blurry mess. So deconvolution, very important. Then pre-processing all the way. You do all of the pre-processing to get the best segmentation. Now I want to show you one of the challenges in segmentation. This is not necessarily good segmentation, but it's a good example to show the problems with segmentation. So I must in some way make a call, what is a mitochondrial structure and what is not? Like I can't work too much with this fuzzy information. It's too difficult to decide when is it now actually fusing and when is it not. So I must binarize it in some point to get, this is a structure, I must consider this structure by itself. But I've got competing goals here. So for example, I've got this strand there. It has this, it's one strand, but it has this low dip in intensity. So it starts bright, then it goes dimmer and it goes brighter again. And that's just inherent. This is actually how it looked in the acquired image. But when I segment, this must be one continuous thing. Now what do I do when I segment, when I basically threshold? I choose where is the best intensity value to consider this foreground or background. So now I must move my intensity value all the way down just so that that is considered one continuous structure. On the flip side, this thing there is most likely on a different level, or this is actually a better example. These two are not touching. They are two distinct separate structures which are close together, but they are not touching each other. You all agree with me on that. They are not fused. Okay, now I've got these two competing goals. I need to be able to deal with these low intensity regions, but when they are close together, then there forms a low intensity bridge between the two, which will then make them seam joined. So just to show you this relatively poor segmentation result, here I've met the one objective, great, that strand is continuous, but those two structures have now become fused. So now I'm going to get wrong results as to where the fusion happened or not. So segmentation is a really, really challenging task, especially for things like this where the actual nuance of touching or not touching makes a massive difference. So in many cases like this, you would maybe even want to use a neural network, which is a little bit more nuanced in doing its segmentation, that you don't rely too much on just histograms and stuff like this. The method I'm showing you now does rely on histograms because everybody might put their own data in this and the neural net is not going to be generalized, so you have to then sometimes just compromise and say, okay, well, this is not a perfect result, but at least let's try to do apples by apples comparison. So this was the initial version of the pipeline. I'm just mentioning things. I'm not going to discuss them in depth, just show some of the reasons for it. So first do deconvolution, then we upscaled it a little bit. The reason for the upscaling is that once we binarize it, it will say this pixel is foreground and this pixel is background. And if these pixels are too close together, as soon as I do the binarization, they are going to appear joined. Where if I first upscale it, I'm just artificially making my blocks larger, that when I segment, then the blocks there are more blocks available to put them as two separate things. So the upscaling just helps to keep things on actually separate, that the blocks, my resolution is not so low that I can't actually do anything with it. I do just a little bit of blurring because of the noise that's in the image. I do contrast stretching to make sure I get the best resolution for ultimately the thresholding to help that to be a bit more consistent. Because different images with slightly different intensities, stretching out the contrast makes choosing a threshold a bit more consistent. Then I used something called hysteresis thresholding, which we haven't discussed. But what this tries to achieve is more nuanced than just picking one threshold and then it's cutting everything off at that point. The hysteresis threshold requires two thresholds. So if you imagine now your histogram, I'm going to just paint it out with my hand. I've got a histogram like this. This is the low intensity, this is the high intensity. I've got my background, there's a bit of a bump there. I've got my foreground. Normal, ideal situations, you can just put your threshold in the middle. But now in our cases, we don't have such a clear separation between foreground and background because I've got all of these extra blurriness. So I would rather say there is a point where I definitely know an intensity above this is definitely foreground. So always include this. And then there's another point where I say everything below this is definitely background. But in this middle region, I'm unsure. So what the research thresholding allows you to do is you set these two thresholds, and then it says, if I found a structure in the foreground that is attached to some pixels which are in this uncertain range, then I will include them as also foreground. Where if the pixels in this uncertain range is not in any way connected to a structure which is definitely part of foreground, I'm going to remove them. So that allows you to have a more nuanced way, and especially with these different competing goals of I've got this intensity dip, but I now have got two structures close together. It kind of solves that problem to a large part for you. Then there were some other steps that I did. I'm not going to go through them. Did some edge detection. The latest version of this method doesn't rely on edge detection. Lots of comparison of matrices and whatever else. Eventually you get to the result. Here's just a bit of an intuitive way of thinking about what it does. I've got two frames here. This is obviously made up frames. If in the one frame I've got one structure, in the next frame there's a little piece separate from that, so this one structure will be associated to both of those structures, then I can say there was a fission event, so I mark it as a red dot there, there was a fission event there. And if I had one structure, or three structures in this case, which then eventually became one bigger structure, then I can say that there were two fusion events that happened there. And then this last one, there was a structure there, and now there's no structure, so I can consider that a depolarization event. I don't know if you can see that, but there's a little blue dot in the middle there. And in order to get it for both fission and fusion, for the fusion case, you go from frame 1 to frame 2. You try to figure out which one structure is associated to two structures. For the fission case, you just go from frame 2 to frame 1, where you say, now which one resulting structure came from which two source structures. So it's the same approach back and forth, but you get the fission and fusion out of the results. Okay, so here are some of the results that we got from this. Applied it to various treatment groups and everything, but this is now a control cell. I think I thought I extracted the histogram as well, but one of the benefits of this method is it can show you over time how the relative fission-fusion frequency or quantities of whatever number of events change over time. So if it's a control cell, you would generally expect the mitochondria to be happily fusing and fragmenting as it needs change, but these should roughly be in equilibrium. And you can kind of see that it stays roughly in equilibrium over time. The depolarization for a control cell should be quite minimal, which is what happened here. And if you had a treated cell, and unfortunately I don't have a slide for this, you would sometimes, and it's kind of tough to catch, but you would sometimes have everything in equilibrium, and then suddenly there would just be a massive spike in fusion, and then it would be fusing a little bit, and then it would again be back in equilibrium. That's kind of like that animation I showed you initially. The cell was nicely spread out, it heavily fused, and then it just, okay, now it's fused. It can't fuse anymore, so then the fusion drops off again. And usually if you see a spike in fusion or in fission, that means there's something wrong with your cell. It's busy dying, or it's not happy. It's under some kind of a stress. Now to just show some examples of what it does here. If I've got, let's say in frame 1 I've got structures that are separate, frame 2 structures that are joined, it shows there's a fusion event. This is a little bit off screen, but if there are structures that are joined, it's not a little bit off the edge, but then clearly it's separated, then it shows that there's a fission event. This is maybe a nicer example. This is a phenomenon known as kiss and run for mitochondria, where you've got two structures, they come close together, then they fuse. So there's a fusion event. You can clearly see that it is actually now one structure. It stays fused for a while, and then after some time, it decides to break up again. So that's the run part of kiss and run. And if you now set up some window around every one of these events, and you say, just check this event over time, did it change or alternate between fusion and fission for the exact same location, then you can pick up these events automatically. So some of the challenges with this, I did already comment on it, but I think it's good to highlight. If you take frame 1, frame 2, a false fusion event would be where there's a structure, it came close, and even this one started to tilt up, but it hasn't quite fused yet. It's maybe starting to fuse, but I can't objectively call that fusion. But because of the low intensity bridge there, my segmentation said that it has been joined, and then I incorrectly marked that as a fusion event. On the flip side, if I had something like this, where again they're not joined, but they're pretty close, so because I need to get this entire leg as one continuous structure, I had to take my segmentation to be quite a low threshold, and then this becomes joined. And now that they're actually moving further apart, the segmentation no longer confused it, and now it says, hey, there was fission, but actually there never was. So there are these challenges, and it's all about down to how did you pre-process and how did you segment your images. The latest version of this tool that I've developed just takes in segmented images. The segmentation used to be brought into the pipeline, and then people were just blindly using parameters without thinking about what they're doing. So I've removed that out of the use of it. You must first segment something, make sure that this is perfect, and then you put it into this method because it's so heavily dependent on that. Another place where it initially failed, it no longer fails in these cases, but if the structure moved too much, if it's a small structure and it moved too much, it was struggling to associate it together with each other, and then it would have said, oh, depolarized, when it didn't actually depolarize. Another issue that it had, which is, again, this is also now resolved in the latest version, these are instructive lessons, I hope, so that if you were to do things, there are some assumptions that you make which might not be valid, and that's what I'm trying to highlight here. So I've got two structures. They clearly fuse together in this third structure in the middle. The green and the red are the before images, and then the gray is the after image. So what I initially did was, it's like, okay, well, I need to somehow determine, I only have the structures, I need to somehow determine where did this fusion or fission happen. So let's just take the best assumption. Well, it seems like it always happens at the closest point between the structures. There was a structure, the closest point between the two structures, that is where it happens. Great, good assumption. Except if something like this happens. Now this was the closest point between the structures, so I said, there's the event, where in reality it fused to a third structure, and there should have been two events. So I missed one event, first of all, and secondly, I put the location in the completely incorrect place, because I assumed that, I just didn't think about this, that this might ever happen. So the latest version of this method, it actually tries to follow a path. It skeletonizes the thing, it essentially reduces the entire structure to a single row of pixels for this entire, a network of pixels, but it's a single line. And then I try to follow this path all the way from the one structure to the next structure, and the only way that I find a path in this case would be through this third structure, and then I would say, oh, I transitioned to one structure, fusion event, okay, I transitioned to another structure, another fusion event. So it's not actually following a path instead of just saying closest point between two structures. This is currently available as a feature plugin. There are a few people here in physiology that have used it for their research. Admittedly, it's not very user-friendly at the moment. Time is always the problem. But yeah, you can download it already. This is an example of if you wanted to use it. There's a lot of parameters that you can provide. Almost all of it you can leave exactly as the defaults. You just need to specify where your path is that you want to save files and where the input file is, for example. But if you want to use it, rather speak to me. It's not currently, like I said, extremely user-friendly. Okay, back to this picture. We have now generated our ground truth. Got a time-lapse sequence, got our ground truth. We can go and train a neural network now. That's exactly what we did. I'm not going to go into the entire thing of neural network training, but usually as it trains, there's a loss that you're looking at. The loss is supposed to decrease. If you look at your, you usually take your training set and split into two portions, a training data and a validation data. The validation set you never show to the neural network. It only trains on the training set, and then you test periodically on the validation set to make sure that I'm not overfitting, I'm not memorizing my training data. Because as soon as you memorize your training data, you stop generalizing. You're not solving this problem in a general sense, but you do very well on the few examples that you give it. So when you see something like this, the validation loss goes nicely down, and then it starts increasing, then you say, that is the point I need to stop training because I'm starting to memorize my training data. So we did that, and then we didn't have, admittedly, that much data to train on, but this is what we get. Now, I want to make a few comments on this, because this is surprising for a few reasons. So a neural network received one image, no time-lapse sequence, just a single Z stack, and then it was able to predict all of these locations. Now, some of them can easily be dismissed already, so if it's below a certain intensity, usually the neural network isn't very clear or sure about that, so we can just filter them out pretty easily. Other cases such as this, where it's also unsure, it decides to put a red dot and a green dot at the same place, so it's unsure, so it just says, something's happening here, but I don't know what, and then we just exclude them. So after we filtered out many of the obvious things that are wrong, and we looked at the ones that remained, and we literally by hand considered each one of them individually and said, in the frame 1, frame 2, the neural network said there's a green dot. Does it make sense? Okay, yes or no? And then we did this, and we got about between 40 and 50% accuracy. So 40 to 50% of the dots that it placed was actually perfect. It was accurate. Many of the others you could say, this is reasonable, but yeah, objectively it's not. Now, remember, the neural network did not see 2 frames. It only saw 1 frame. So it accurately predicted 50% of the locations purely based on the morphology of how the sample looked at the moment. And also another thing is that event that might have got be incorrect, that was only incorrect necessarily for this next 10 seconds for the next frame. But in some cases, we looked 1 frame on, it's like, oh, it was going to fuse. It was just like not within the time frame of 1. So the insight more is not that you should use this on a single frame. This is actually our conclusion is it's too inaccurate to use on a single frame. You get about 50% accuracy. So the counts don't mean that much. But it is indicative that the morphological structure gives a very strong predictor as to where fission fusion is supposed to happen. So the dynamic remodeling of the cell, highly dependent on the current morphology of the cell. So that was kind of an interesting insight. Okay, now just shifting on to another method. Some of the things that this is a Nature Methods paper. One day when I'm old and wise, I might also get a Nature Methods paper. But they did something very similar to what I did. It's just that they focused more on the migration of mitochondria, and they tracked it in an interesting way, which I think you might understand most of what they did by now. But they don't get the location as to where the events actually occur. So there's some space for that method of mine, which is called mitochondrial event localizer, or MEL for short. But this one does something similar. So what their goal was they first tried to calculate for every single mitochondrial structure. They calculate different parameters. This is now volume, solidity, major axis, minor axis, surface area. So you remember those measurements that we've discussed yesterday. So they get for every structure a certain set of parameters, and then they say, I'm going to call every structure, this is now a new track essentially, and then let's look at the next time point. Let's calculate all of these parameters again. Now let's try to match these tracks together. So over time, this is not a very easy to understand plot, but basically it says, looking at all of these quantities, from the 1 frame to the next frame, which structure is most likely the same structure as what I had before? And then they start matching structures individually from 1 frame to the next frame. And then if you've got 2 tracks over time that were 2 tracks, and then in the new frame they become 1 track, then they obviously fuse. If you had 1 track and then suddenly out of the same region another track just spawned, then you say that is a fusion event. So it's a similar mindset, but it's a slightly different approach to what I took. I'm still to validate exactly how my method compares to theirs. I think in some cases mine is better than theirs, in other cases possibly not. I haven't fully investigated this, but that's the approach what they're doing here. So one very nice thing of tracking these tracks over time is that you can actually get the speed of mitochondria. So mitochondrial structures, how rapidly do they move around? And often, I've seen this visually, if they're on the periphery, they can really dart very quickly from one place to another place. If they're inside this network, they're usually more contained. They wouldn't jump around as much. And they could then quantify this, or in general, the movement speed of mitochondria under different conditions. They could also calculate these ratios, fission-fusion ratios. They called it the normalized fission rate, which is normalized by, I think, the number of events, not normalized maybe by the events at 0. I'm not exactly sure with what they normalized here. But the idea is that they get the rate of fusion, and then over time, so here you can see this thing is clearly biased towards fission. It's increasing compared to what it was initially. And the fusion stays more or less the same, similar to the plots that I got. The big difference here, however, is that export or demonstrate or visualize where these events happen. So where my thing generates a visualization, you can see, oh, it's in this region of the cell. So let's see what is happening in that region. So okay, there might be some more abundance of autophagosomes in that region. So therefore there is more whatever happening. You can start to make more interpretations because you've got location information, where in their case you'd only have the numbers information. But you also have this visualization to see how the different things moved around, and it's basically just a large list which they plotted out here. Yeah, so that's my talk on mitochondria. Any questions? Any comments?